Hi everyone. Today we will be finishing our discussion on polysaccharides. Specifically, we will be talking about heteropolysaccharides, and then we will also talk about glycoconjugates briefly. We will go over these in more detail in our next video. So heteropolysaccharides are polysaccharides that are made up of two or more different types of monosaccharides. And the first one that we will be looking at is peptidoglycan, which reinforces the bacterial cell wall. And it is made up of alternating N-acetylglucosamine and N-acetylmuramic acid residues. So as we can see here, the N-acetylmuramic acid links to short segments of amino acids, which we will call peptide crosslinks. And so here is a more simplified diagram. The green hexagon is the acetylmuramic acid, and it links to the peptide crosslinks. It has different amino acids like alanine, and then these peptide crosslinks will then link to the acetylmuramic acid of another peptidoglycan chain. So in the bacterial cell wall, the peptidoglycan chain will, be in this sort of conformation, and it stretches from left to right with peptide crosslinks between the acetylmuramic acid. So M stands for acetylmuramic acid. And the glycosidic bonds confer strength on peptidoglycan in the X direction, whereas peptide bonds confer strength in the Y direction. The enzyme lysozyme kills bacteria by hydrolyzing the beta-1 to 4 glycosidic bond, so that is the bond between the N-acetylglucosamine and N-acetylmuramic acid. And the enzyme is found in human tears, where it can presumably defend ourselves against the bacterial infections of the eye. And yeah, so basically the structure of peptidoglycan reinforces the bacterial cell wall. Moving on to glycosaminoglycans, they exist in the extracellular matrix of animals, and they are made up of alternating uronic acid and amino sugar units in red and blue respectively. Keratin sulfate is an exception. It doesn't have uronic acid. And many has sulfate groups in green, as we can see here, except hyaluronan. Because the sulfate groups and carboxylate groups are all negatively charged, the glycosaminoglycans form an extended rod-like helix to maximize the separation of these negative groups and thus minimizing repulsion. So here we have the 3D structure of heparin. Hyaluronan acts as lubricants of synovial fluid of joints, and it is also a part of the extracellular matrix of cartilage and tendons. By the way, these glycosaminoglycans, they exist in a lot of connective tissues, like these. Other glycosaminoglycans include chondroitin sulfate, keratin sulfate, and heparin sulfate. The reason that they are grouped differently from hyaluronan is because they are different from hyaluronan in three respects. The first being that they are generally much shorter polymers. The second being that they bond covalently to specific proteins called proteoglycans, which is a type of glycoconjugates that we will talk about a little bit in this video, and also more extensively in the next one. And the third being that they have at least one monomer that's different from that of hyaluronan. So chondroitin for sulfate, or chondroitin sulfate in general, strengthens cartilage, tendons, ligaments, heart valves, and walls of the aorta. Keratin sulfate exists in the horn, hair, hooves, nails, bones, and cartilage of animals. Heparin sulfate are they contain variable non-random arrangements of sulfated and non-sulfated sugar, which allows for specific interactions with proteins, including growth factors, ECM components, so extracellular matrix components, and enzymes and factors in the plasma of blood, such as thrombin. Um, so you can think of these like sulfated groups that we just mentioned as sort of, I don't know, they might be like analogous to the base pairs in DNA, and the sequence of these sulfated residues will allow for it to interact specifically with proteins, with and like ligands in general. And heparin is a highly sulfated intracellular form of heparin sulfate. And it is produced primarily by mast cells, MAST. They are a kind of immune cell, and its physiological role is unclear. So you might be the one to figure out what heparin does. Yeah, let's take a moment to pause before we move on. So we will be talking about glycoconjugates briefly, but before we do so, let's remind ourselves of what we have learned so far. Polysaccharides, so carbohydrates, they serve structural functions. They provide support in the extracellular matrix of animals, or like the plant cell walls, the bacterial cell walls, and also chitin exists in the fungal cell walls. So they can provide structural support, and also they can act as energy storage, right? So glycogen stores glucose in animals and bacteria, while amylose and amylopectin stores glucose in plants. So that is what we're familiar with, right? And glycoconjugates, they are they might be a newer concept, and they have a lot of different functions in the body. So it's like a leap forward in our knowledge of carbohydrates. They serve in cell communication, protein labels. They serve as protein labels for transportation, localization, or destruction of these proteins, and they recognize as extracellular molecules or parasites, and they're involved in blood clotting, immune response, animal development, and wound healing. So there's a lot going on. And as you can see, their structure sort of fitting their function is like highly diverse. Glycoconjugates are basically carbohydrates covalently linked to lipids or proteins. They have three types, the first one being proteoglycans, which are molecules in which sulfated glycosaminoglycan chains covalently link to proteins. The negatively charged groups can bind to extracellular proteins. So that's the heparin sulfate that we're talking about. Although like chondroitin sulfate and other glycosaminoglycans can also be covalently linked to proteoglycans. Glycoproteins have one or several oligosaccharides of varying complexity joined covalently to a protein. So oligosaccharides are basically polymers that are shorter than polysaccharides but longer than disaccharides. As we can see, they are very diverse and thus can bind to specific carbohydrate binding proteins called lectins. And glycolipids are membrane components in which the hydrophilic head groups are oligosaccharides. So they're sort of like phospholipids, but not really, because the polar part is oligosaccharide fragments. Glycosphingolipids are a class of glycolipids which can also be recognized by lectins. So yeah, that's it. See you in the next video where we will be talking about glycoconjugates in greater detail.